Welcome to today's explainer. I'm super excited you're here because today we're going to literally map out the exact mechanical relationship between your microscopic and visible alleles and the highly visible physical traits they actually build. We're going to use the screen as our interactive whiteboard to strictly follow the flow of information, from the genetic code right up to the living organism, completely decoding how inheritance works along the way.
So, did you know that the exact same mechanical process determining the color of your eyes also dictates how tall a plant grows in your garden? It's true. Whether we're looking at complex human traits or just the simple height of a garden pea, both rely on the exact same underlying genetic principles. By the end of this explainer, you're going to have the visual tools to calculate and predict these biological secrets for yourself.
Here is our roadmap for today. We'll hit the genetics of traits, define purebreds versus hybrids, map out Mendelian monohybrid crosses, predict F2 generations, separate genotypes from phenotypes, and then wrap it all up with a quick recap.
All right, let's jump right into Section 1, the genetics of traits, where we discover the rules of inheritance.
Moving straight into Section 2, purebreds and hybrids. Let's decode the letters of DNA. To really get this, we need to zoom way in on the micro-level letters of our genetic code. See that definition popping up? Homozygote. In everyday language, you'd just call this a purebred. It's an organism that has two absolutely identical alleles for a specific trait. Notice the call-out letters on screen? Capital T, or lowercase t. The absolute key takeaway here is that perfect identical pairing. Now let's split the screen. On the left, you've got our purebred homozygote. But look over on the right at the heterozygote, which you probably just know as a hybrid. Notice how the hybrid has a mismatched pair. We've labeled it right there, a dominant capital T paired directly with a recessive lowercase t. Slapping that dominant capital letter next to a recessive lowercase one completely and fundamentally changes the underlying genetic makeup of the organism. Which brings us to Section 3. Mendelian monohybrid crosses and Mendel's famous pea plant experiments.
A monohybrid cross simply means we're tracking one single characteristic at a time. This exact method was the whole foundation of Gregor Mendel's work. Let's calculate this step by step together on the grid. We've got a 2x2 Punnett square here. We're crossing a purebred tall plant, look at the callout, capital T, with a purebred dwarf plant, labeled lowercase t. Now watch the arrows. As we distribute the parental alleles into the grid, the capital T's flow straight down from the top. The lowercase t's flow directly across from the side. And boom, as they converge, the result stamps perfectly into every single empty box. Capital T, lowercase t. Literally 100% of this first generation, what we call the F1 offspring, become heterozygote hybrids. And since that capital T is dominant, every single one of these plants is going to physically grow tall.
Okay, Section 4, predicting F2 generations. Time to calculate some genetic outcomes.
So, we know our first generation are all hybrids, but what on earth happens if we cross them with each other? Let's treat this like a strict flowchart. Step 1, highlighted right here. Place our two F1 hybrids, capital T, lowercase t, on the top and side axis of the square. Step 2, we pull those alleles apart, getting them ready to drop. And Step 3, combine them. Drop the top alleles down, slide the side alleles across. See how the top left box gets two capital T's? The top right and bottom left both grab a capital T and a lowercase t. And down there in the bottom right, it receives two lowercase t's. By physically tracing those directional paths, we've just precisely mapped out the genetic outcomes of the F2 generation. Let's quantify that data and look at the proportions on this chart. Exactly 25% of the offspring are purebred tall, TT. A massive 50% chunk right in the middle is hybrid tall, TT. And that remaining 25% slice, purebred dwarf, TT. This right here gives us our golden Mendelian genotype ratio, 1 to 2 to 1. Now let's zoom out. Here's where we bridge the micro with the macro, shifting from that invisible genotype ratio into the highly visible physical phenotypic ratio. Because both the 25% TT and the 50% TT offspring have at least one dominant capital T, those plants are going to grow tall. That leaves only the 25% TT section to actually manifest as physically short. So, looking at the whole macroscopic picture, our physical ratio is exactly 3 tall plants for every 1 dwarf plant.
All right, let's run just one more calculation to really lock this in. This time we're crossing a heterozygous tall plant, capital T, lowercase t, with a homozygous dwarf plant, lowercase t, lowercase t. Watch the paths of the alleles dropping into the grid again. Look at that. It splits straight down the middle. 2 boxes read TT and 2 boxes read TT. This visually proves that this specific cross gives us an exact 50-50 split, 50% tall, 50% dwarf, a perfect 1-to-1 ratio.
Moving on to Section 5, genotypes versus phenotypes. Let's make sure we avoid some really common mistakes.
Now that you've seen the math, we absolutely have to make sure you never confuse the underlying mechanics with the final physical product. Look at the split screen. On the left, we've got the genotype. That's the micro-level invisible genetic makeup, the actual TT, TT, or TT alleles sitting deep inside the cells. On the right, the phenotype. That's your macro-level observable physical trait. It's the actual tall or dwarf plant that you can see and touch. And hey, here is a massive misconception we need to bust right now. Just because an allele is dominant in a genotype does not mean it's automatically going to be the most common phenotype in the wild. No way. Dominance only dictates how an allele behaves when it's paired with a recessive one. It tells us absolutely nothing about its overall population frequency.
Finally, Section 6. Quick recap and tips. The absolute essential takeaways for your exams. Let's do a rapid-fire summary to lock this all in. Number 1, homozygotes are identical, purebred alleles, while heterozygotes are mismatched hybrids. 2, monohybrid inheritance focuses strictly on tracking one single trait at a time. 3, always use the step-by-step flowchart of a Punnett square to mechanically predict your outcomes. 4, never ever confuse the invisible genotype code with the physical phenotype trait. And lastly, here is your golden exam tip. Always double-check which alleles you've labeled as dominant and recessive before you map them into your square. I mean it. Mixing up a capital letter with a lowercase letter will literally ruin your entire calculation.
So, I want to leave you with a bit of a thought-provoking question. If dominant traits can so easily mask recessive ones, like a tall trait completely hiding a dwarf one, what invisible recessive traits might you be carrying around in your own genotype right this very second? You are literally a walking, breathing combination of dominant and recessive codes, just waiting to be mapped out. Thanks so much for joining me for this explainer, and until next time, keep questioning the mechanics of the world around you.